Today we are going to discuss about the topic the amino acids. Before going toward the amino acids, we need to know about what is an amino acids. We all know about the biomolecules. Biomolecules mean they are the molecule that is found in our system. Therefore, some of the important biomolecules are carbohydrates, amino acids, nucleic acids and proteins. Here we are going to discuss about one of the main and important biomolecule, that is amino acids. And coming to their structure, actually we all know that amino acids are the building blocks of proteins. That is, proteins are composed of amino acids. They are the polymers of amino acids. That means proteins are the polymers of amino acids, and these amino acids are linked by the peptide bond for the formation of a particular proteins. And there are around 300 amino acids in nature, but we are going to most important one is 20 amino acid that is found in biomolecule or in humans or in organism. Therefore, we are mainly highlighting, or we are going to discuss about this 20 of the proteins or 20 of the amino acid that is occurring in proteins. Repeating, the amino acids are the building units of proteins. Amino acids, they are the biomolecules. They are the building units of protein, and these proteins are polymers of amino acids. And there are around 300 amino acids, but in proteins there are only 20 amino acids. That is why we are dealing with the 20 important amino acids that is found in protein. That's found in proteins. Coming to the structure of amino acids, here we can see the main important structure. We can see the carbon. Carbon is the main important compound in the proteins. Here, carbon atom next to COH is known as the alpha carbon atom. This is the alpha carbon atom, that is the carbon atom next to COH. That is the carbon atom next to COH, that is the alpha carbon atom. And these four groups are amino group, and this alpha group is valencies. Alpha carbon valence is fixed by amino group. One is the amino group, this is a carboxylic group, and one is a hydroxyl group, and this R represent the side chain. By changing this side chain, we will having different type of amino acid. So the basic structure of amino acids contain the middlemost is alpha carbon atom, and this alpha carbon atom's valence is filled by carboxylic acid group, amine group, hydroxyl group, and this R represent for side chain. Depending upon the varying side chain, we are having different amino acids. Then coming to the peptides and proteins, that means amino acids combine together for the formation of the proteins. What is the difference between peptides and proteins? We already told that there are 20 important amino acids. These 20 amino acids are linked together by peptide bonds, and if that chain contains 50 amino acids, that is, whenever these 50 amino acids are binding together, they are known as the peptides, while those containing greater than 50 amino acids, they are known as the proteins. 50 amino acids, up to 50 length amino acids, we are known as the peptides, and whenever there are large number of amino acid group, they are known as the proteins. Coming to the peptide bond formation, that is how that particular bond is formed, which bond the peptide bond is formed. The carboxyl, we have already seen the structure of the amino acid, that is, this is the carboxyl group and this is the amino group. In case of the peptide bond formation, the carboxyl group of amino acid and one molecules of water is removed from the carboxyl group of one amino acid and amine group of other amino acid. That is, here whenever we are discussing about the protein, we already told that there are large number of amino acid molecules packed together with the peptide bond, and this peptide bond is formed by the removal of water molecule. Therefore, that is a dehydration reaction, the removal of water molecule by the combining of the carboxyl group of one particular amino acid and amine group of other particular amino acid. And whenever two amino acids are bind together, they are known as a dipeptide. By the same way, dipeptide can then form a second peptide bond with the help of a third amino acid for the formation of tripeptide. Coming to their structure, here we can see two amino acid. This is one of the amino acid, this is another amino acid, and This is another amino acid. Here we can see H2O, that is the OH group from the carboxyl group and hydroxyl group from the hydrogen group from the amine group is binding together for the formation of water molecule, for the removal of the water molecule, and by the removal of the water molecule this will be linked like this figure. That is, here we can see 3 amino acids are linked by peptide bond, and this peptide bond is formed by removal of water molecule from the carboxyl and amine groups between this structure, and this is known as a peptide bond. And this is all about the structure of the peptide bond. Once more, in peptide bond is formed by the removal of water molecule by removing hydroxyl group from the carboxylic acid and hydrogen group from the amines. Now coming to one example, here we are taking the example of alanine and glycine. We all know about the alanine and glycine. Glycine is the simplest amino acids as well as the alanine. Here this one is alanine and this one is the glycine. Here we can see carboxyl group, hydroxyl group, and here what is the difference between glycine and alanine is that alanine is having CH bond on SR, and in glycine that is the hydrogen molecule. That is the main difference between alanine and glycine. Now we are going to discuss about how the peptide bond is formed between alanine and glycine. And here this is the peptide bond between the alanine and glycine. Here we can see the hydroxyl group from the carboxyl of the alanine and hydrogen group from the amine group of glycine is removed by the formation of water, that is H2O, and they are linked for the formation of the peptide bond. And through this way the peptide bond is formed. Here that is one of the example is alanine and glycine. Here alanine. In alanine what happens? The carboxyl group of alanine's hydroxyl group is removed and the glycine's hydrogen group from amines is removed, and they are packed together and they are linked for the formation of the peptide bond. That is, 2 alpha amino acids are by peptide form for the formation of the dipeptide. This is the same figure but representing another amino acid, that is the amino group. Here we can see one amino group and their carboxyl group, and they are bound together for the formation of the peptide bond, and peptide bond is also known as the peptide linkage. Coming to their, this is all about the structure of the amino acid. That is, we already deal with what are the molecules found in the amino acid, that is carbon, hydrogen, oxygen, and we already deal with the carboxyl group, amine group, hydrogen and side chain. This is all about the structure. We are going to discuss about the structure of protein. Already the proteins are formed by the amino acid. They are the polymers of amino acids. Now we are going to discuss about the structure of protein. We all know that the proteins are the building blocks of our body, and these proteins are again built by the amino acids. Now coming to their structure, there are mainly 4 levels of protein structure, and they are primary structure, secondary structure, tertiary structure and quaternary structure. First one we are discussing about the primary structure. First one, the primary structure means primary, that is the basic structure. Basic means we already deal with on the last slides, that is about the amino acids. Individual amino acids combine together for the formation of the peptide bond, that is an unbranched or lengthened bond, that is known as the primary structure of the protein. And the primary structure of protein is determined by inherited genetic information. That means the proteins are formed by translation, that is a genetic information, that is DNA replication, transcription and translation. These are the main central dogma of our life, where replication means conversion of DNA to DNA, after that DNA to RNA, and from that RNA to protein, that is known as a translation. Therefore, the translation is the main synthetic pathway of the protein. That is why the primary structure of protein is determined by inherited genetic information. And at one end of amino acid with a free amino group. Coming to the structure of the protein, we already told that 50 particular amino acid is known as the peptides, and more than 50 amino acid it is known as a protein. Coming to the primary structure, there are two ends in the protein structure. First end is carboxyl group and other end is the amine group. Here is the structure, that is, here we can see one and amino group, and last one is a carboxylic, and in between there we can see total number of 129 amino acids. In this figure we can see 129 amino acids. This is the primary structure. Again, just to elaborating this one, this is another primary structure. Here we can see just sulfur bond, disulfide bond. We can see these are all the different type of amino acid. This is the amino and last one is the carboxylic and. So this is all about the primary structure. Coming to the secondary structure. Secondary structure means the bond formation, that is mainly the results from hydrogen bond formation between amine group of peptide bond and carboxyl group of another amino acids. There are mainly important two types of secondary structure. They are alpha helix and beta sheets. There are alpha helix and beta sheets. Coming to the alpha helix from the name itself, it is a spherical structure or helical structure. Spherical, coming to the spherical structure, it is more important, or we can tell it is a helical structure resulting from hydrogen bond between one peptide bond and the fourth one. That is, in primary structure, we already told that this is primary, that is amino acids are bind linked together for a lengthened one. In secondary structure, there will be a hydrogen bond between amino acids, between the first amino acid and other fourth amino acids. The first amino acids hydrogen bond with fourth amino acids, and that is the alpha helix, that is having a helical structure. And coming to beta sheets is also another secondary structure, but difference is that in beta sheets a hydrogen bond will be between intermolecular hydrogen bond is formed. That is, in case of alpha helix, they are intramolecular hydrogen bond. That means a hydrogen bond between first amino acids and fourth amino acid. But in case of beta sheets, we can see hydrogen bond that is intramolecular between the polypeptides. The hydrogen bond between adjacent peptides is formed in beta sheets. They are linked together by hydrogen bond between the hydrogen atom of one chain and other chain. The hydrogen bond between two chain, they are known as the beta sheets. Here we are elaborating the alpha helix. Alpha helix means it is a polypeptide backbone that is wound around an imaginary axis. Coming to their structure, we want to first imagine an imaginary axis, and through this axis the alpha helical structure is wrapped here, and the first amino acid is having an hydrogen bond between the fourth amino acid like this. This is the imaginary axis, and these proteins or polypeptides are wrapped around. This is a 3D structure of the alpha helix. Here we can see carbon, hydrogen, oxygen, nitrogen and R group. And this is all of the alpha helix. And in alpha helix, there are certain amino acids that destabilize alpha helix. Destabilize means whenever there is a presence of D amino acids. D means dextra amino acid. We already know about the D amino acid and L amino acids. Whenever there is a formation, there is D amino acids in that particular chain, it will destabilize the alpha helix, or it will disrupt that particular structure of the alpha helix and contain long blocks of, if that particular amino acid contain long blocks of glutamic acid, lysine and arginine, it also destabilizes that particular structure, as well as the threonine, leucine, etc. Okay, and proline and glycine. Proline and glycine are helix breakers. Whenever there is a formation of proline and glycine amino acid, it will break the alpha helical structure. Coming to the beta sheets means that is the hydrogen bond between the chains or the sheets. It is sheet-like structure. We already know about the web of the spider web. They contain the beta sheet-like structure. Therefore, that give the structural property, and it has hydrogen bond between in alpha helix, and in beta sheet they are having hydrogen bond, but the main difference is in alpha helix that the hydrogen bond is in between the chain, but in case of beta sheet the hydrogen bond is formed between the chains, means between the chain, but in alpha helix it is between in single chain, the bond between the single chain. And this is all about the beta sheets. Coming to their structure, there are mainly two types of beta sheet. They are anti-parallel beta sheets and parallel beta sheets. Here the anti-parallel, from the name itself, from the anti-parallel, that is one strand is for this direction, another strand is in this direction. That is why it is known as anti-parallel sheets. And in parallel sheets, it is having a single direction. That is the main difference between parallel and anti-parallel beta sheets. And this is all about the secondary structure. Coming to the tertiary structure, it is determined by variety of bond formation. Secondary structure means there are different type of bond formation. They are hydrophobic interaction, electrostatic interaction, disulfide bond, ionic bond. These are all the interaction found in the tertiary structure. And this is the structure of the tertiary structure. Here we can see the disulfide bridge between the chains, ionic bond and hydrogen bond, hydrophobic interaction. Tertiary structure is important for the interaction. These are all the different interaction found in the tertiary structure. And coming to this last structure, that is the quaternary structure. After the quaternary structure is formed, the protein is having a particular functional protein. Quaternary structure results from the aggregation of two or more polypeptide units. Two or more polypeptide units aggregate together for the formation of the quaternary structure. In case of hemoglobin, we all know the hemoglobin is a protein found in blood, and this hemoglobin's quaternary structure contains two alpha polypeptides and two beta polypeptides. That is, they are having four polypeptides in here. Their structure, that is the quaternary structure of hemoglobin. And these polypeptides are aggregated together for the formation of the quaternary structure. One of the examples is collagen. We all know collagen is a fibrous protein. It contains 3 polypeptides. In case of hemoglobin, it is a globular protein with 4 polypeptides, and insulin contains 2 polypeptides. This is the structure of the quaternary structure of the collagen. And hemoglobin, here we can see, we already told that hemoglobin is having 4 chain, 4 polypeptide, 2 pink, and here the color represent pink represent the alpha chain, and the orange color represent the beta chain. These are the 4 polypeptide found in the hemoglobin. This is the overall structure of the protein. We are discussing about the protein structure. There are mainly 4 structures are there. That is primary, secondary, tertiary and quaternary. The first one is a primary structure, that is a simple amino acids just linked with the peptide bond. Coming to the secondary structure, that is the alpha sheets and beta plate, alpha helix and beta plated sheets. Here we can see different hydrogen bond formation. Coming to the tertiary structure, there includes different type of interactions such as hydrophobic, ionic bond, disulfide linkage, etc. And last one, that is a quaternary structure. It is an aggregation of so and so many polypeptide. And this is all about the protein structure. And thank you. So today we have discussed about the amino acids, their structure, how proteins are formed, and last we have discussed about the protein structure, that is different levels of protein structure. Okay, thank you.